Yeah, but first I'll introduce you.
He's asking.
Are you president?
Do you want
Good
us to sit?
afternoon.
I'll sit right here.
My name is Field Horn. I'm president of the Saratoga County History Center, and I am very pleased to welcome the Heritage Hunters here today to the center on behalf of the entire Board of Trustees. It's a natural partnership between a county history center and a county-based genealogical society, and we have for your use a research library. And actually, most of it belongs to Heritage Hunters, but we are housing it. We also have a very large manuscript collection with many, many, many family records that are not in the library. They are in archival storage. And finally, we have a huge photograph collection. Soon enough, these will be accessible through various finding aids, and you'll be able to look for people that you're searching for. Despite the fact that you don't see me much, I have been working in genealogy for 60 years. I published my first small genealogical book 60 years ago. It's very bad. It really is very bad. Another one in 1970, and then beginning in 1974, three. My father's family, my mother's family, and last month, the family that I have my first name from. And this one is 365 pages hardbound, so I went for broke with it. Our objective here is to return this institution to full stability within about 3 years. This is going to require increasing our budget considerably. As you probably know, we've had an executive director. Funds were not adequate to maintain that position. At the present time, we have a half-time educator, and now we have a museum assistant. This is very good, but as you can imagine, employing an executive director is likely to increase our budget by about $60,000. So we have a long way to go, and I will therefore make my pitch that there is no better way for you to help us move toward this goal than to become a member of the History Center. This brochure is in various places, but most easily found on the reception desk in the middle of the first floor. Memberships start at $25 and go up from there. We have about 345 members now, a very great increase from 145 in 2018. When I left here as executive director in 1985, we had 550, and we need to go up to probably 600 at least. So please give that serious consideration. And now I will turn the tricky microphone over to your president.
Well, welcome everybody. I'm Dave Beck, president of Heritage Hunters, and I want to thank Field Horn and the Saratoga County History Center, aka Brookside, for hosting us today. It's a terrific venue for us to take advantage of, and I've been traveling a lot, so everything that's going on here today, you have to thank people like Ginny, who's going to introduce our speaker, and the other members of Heritage Hunters. And when you're in the process of joining the History Center, please join the Heritage Hunters as well, because we have about 250 members around the country, and when we were on Zoom, many of those people could tune in. We want to keep the programs going well as they are. So without any further announcements from me, I'll hand it over to Ginny, program chairman.
Site for meetings next month. So on May visiting the Marshall House sometime this summer. The date hasn't been turned up yet. But it's privately owned, and they're very welcoming to have us visit them. So that should be announced in a forthcoming newsletter. And now I'd like to introduce our guest speaker, Toby Kirschman. Toby is a DNA analyst and also a genealogist. She's one of our members, and we're very fortunate to have her here today. She worked in California for 10 years with law enforcement, was involved in some famous cases. And she also has worked in Albany, and she has started her own company, genealogical.com. And she'll be talking about her work with DNA.
Okay, thanks everybody. I think I'm mic'd up. Can you hear me okay? Good in the back. All right, great. Thank you for the lovely introduction, Jenny. And a few housekeeping items real fast. Everybody in Heritage Hunters got our last newsletter probably a few weeks back. And then I wanted to draw your attention to a couple pages, actually just one right now. There's a DNA interest group that we are thinking about setting up here in Saratoga County through the Heritage Hunters. And so as I give this talk today, just think about that. Think maybe if you might want to join, because so many more people are looking at their own DNA today. It's becoming almost common, right? You know, so if you're finding that for your case, yourself, your family, and you want to look into some mysteries, then I think that there are going to be a few people just like you, and it's time to kind of put a name and a day of the week to that and put some energy behind it. So I look forward to you guys reaching out to me. That's on page 19. So that's my plug for the DIG, DNA Interest Group through Heritage Hunters. I also have business cards here if anybody wants one a little bit later on. So this is my company, DNA Investigations. I've been running this company now for a year and a half, and I live in Saratoga Springs. So this is my first talk live here. I'm so excited since all this COVID stuff. But I really do feel you can really help, you know, globally, you know, across states, internationally, you know, fight crimes and stuff like that. But you can also have a huge impact in your town and in your county. And I really feel passionate about that. So I'm happy to be here today. And I'm really happy to have all of you here too. And together we will move forward to make our area a little bit safer in a new way. So that's the thing, the little interest drop. Hopefully I have your attention now. So what I'm going to be talking today about is genetic genealogy. And that's what I do. Okay, a little bit more of my background. I was born and raised in California. And I would come out here and I saw Lake George and fell in love with it. My husband is from New York, and I knew I could live here. And so when things started burning in California, we were like, oh, do you want to move? And everybody was like, yep. So here we are. Saratoga Springs. And earthquakes. And you can't buy a house. And it's just, yeah, poor California. But so I love it here, though. It's great. And I've adjusted and happy as can be. But before I left California, I have this, you know, kind of track record of being in the sciences and healthcare, and so I just wanted to introduce you to that a little bit because it does kind of matter. Sorry, I'm going to move things around just a little bit here, make it easier for me. I wonder if you can see this. Oh, good. See, it's my laser pointer. So, yes, I did spend 10 years at the California DOJ DNA laboratory, and that was what I just did before I came right here and moved here in 2017. But even before that, I went to college at UC Berkeley, so I have a degree from there, and then I went to Humboldt State University and got a Bachelor of Science there. And so I worked in a hospital, and then I worked in biotechnology, and then I finally got my last degree at UC San Diego. And I was still working in biotechnology, and then I had a friend say, why don't you try forensics? And so I did. And I really liked that, but then Saratoga Springs was calling, and I moved here, and I was like, well, I don't know if I'm going to do forensics anymore. And then right as I was here in 2018, the case of the Golden State Killer broke. And back in California, I had worked on that at the laboratory. And I have a few slides on that later. It's when I get really passionate. But we finally we solved the crime, finally solved it, so that's good. But at any rate, I have all of this background in California. I came here and started working as a forensic scientist at the Forensic Investigation Center in Albany. But then COVID hit, and we were starting to do genetic genealogy, and everybody just went home. And I said, this is a great time for me to do what I really know that I want to do, which is genetic genealogy. So that is the background, and here I am. So far, so good. Okay, so now I have, just to give you kind of a warning, if you've got to hop out and use the facilities or get a drink, please feel free to do that, because I have a chunk of slides, and you really have to have your thinking cap on, because I jump around to a lot of different things, from science to policy to genealogy to history, right? So if you need a break, please take one. But I will have a few slides on science, a couple slides on DNA, and then a few slides on law enforcement, a little bit of the history of how law enforcement started using DNA to solve cases. And then at the very end, I talk about genetic genealogy, and that's when I bring in genealogy and history. So anyway, we'll have natural breaks through all of this, and so raise your hand if you have a question. I welcome that. It's no big deal. Otherwise, at the end, we have a lot of time too, and there will be room for discussion. Okay, so you can do this. Couple of DNA slides. Just clear your mind. And this is really, really what you do need to know, though, moving forward. It doesn't matter what age you are or where you work. Everybody is talking about DNA. Everybody knows a little bit. Okay, so here are the few things that I think are really important. So DNA stands for deoxyribonucleic acid, but you never have to remember that, because everybody just says DNA now, and everybody knows what you mean, so that's great. But inside the DNA molecule, you'll find 4 nucleotides. They're adenine, thymine, guanine, and cytosine, but you don't have to remember that either. You can just remember A, T, G, and C, right? But that's the important part for talking about heredity, inheriting genes, passing them down from grandfather, to father to son. This is what we're talking about, the order of the AT and the G and the C. And that specific order is different for every single person, and that's what makes us all very unique. So that's it. So now you know about DNA sequencing, really, and inheritance in a nutshell. You didn't know it was so simple, right? But anyway, it's the tricky sequence of those molecules. A, adenine reaching across to thymine and kind of holding hands, and then cytosine reaching across to guanine, over and over again in whatever pattern it wants, right? Whatever pattern the DNA wants, and that's what makes everybody totally unique. Also, the chunks of DNA, I'm going to be using that word chunks in this talk today. The chunks are very important. They're also called genes. And so, almost everybody knows what, a gene is these days, but it's, these specific chunks of DNA where the sequence is exactly the same, and that's what's passed down from grandfather to father to son, like that. So it's these sequences, these blocks or chunks that are handed down, and that's what heredity is. Also, before I get off of this slide, one thing that you do need to know is that DNA is a robust structure, and that's and we have those outer rungs to thank for that, shown here in red and green. And so these backbone molecular structures of DNA allow it to be, first of all, in your body in an aqueous environment. So it's kind of wet, right? Your cells are alive, and it's kind of wet, inside, but floating through blood, whatever, right? And it allows these really hardcore structures keep the DNA solid, but also the cool thing for my job and for historians and genealogists is that once you have DNA on something, like my cup here, and then it leaves my body, it goes onto the cup, and then it goes down here for 50 years or whatever, right? We find bones in the desert. We have a piece of evidence from a cold case that's 50 years old, right? It's the cup. We can absolutely find out who is on that cup. I can tell you who drank from the cup. And we can do that today because of sequencing. Because now the technology exists for us to take a really good look at the ATGC lineup in whoever left this cup and tell you exactly 50 years ago who drank from it. So that's what we're doing today, and that's what's kind of capable. Okay, and this is the last scientific slide, I think. So you guys are becoming experts really quickly. So this is a human cell right here, and it's important to know that the DNA is housed in the nucleus of a cell. It's very protected. The cell of a body knows that DNA has to last, can't be messed up or anything like that, right? So it has its own little house. All of the industry happens in the other part of the cell, the bartering and the transferring in and out of all this kind of stuff, and the upkeep of the cell, in its production. That's all going around outside the nucleus, but nothing happens to the DNA. It's sheltered inside and protected. So also what's important here is that DNA is supercoiled into, which almost everybody knows about, called chromosomes right there. And I mentioned before those chunks, and you can actually see them represented here by these black spots, right? So these are the stretches of DNA. And if you took that first black chunk on a chromosome and you unwound it and made it linear, you would have the picture of DNA that we looked at in the slide before. And inside, you'll have the AT, GC, and it's whatever combination your mother wanted it to be. So that's how it lays out in your body. And so it's the chromosomes that house the DNA, and it's the nucleus that shelters those chromosomes. Ta-da, you're all done with molecular biology. Congratulations. The next 3 slides have chromosomes. And I mentioned that I worked in a hospital for a while. It was during the 90s, early 2000s. And we still did something called karyotypes when we feared that there was a genetic problem going on with an individual. And so this is just a karyotype that I worked in the laboratory. And a doctor would say, gee, I wonder if what's wrong with this patient is that they have maybe a genetic anomaly or something going on there. So they would order a karyotype. And then I just thought it was so beautiful. You could just look at your chromosomes. And I thought, wow, people are worried about their social security numbers, but this, man, that really just tells everything about you. But what I like about this, it's old-fashioned, and I don't think the medical community uses karyotypes anymore. I'm sure they have something, so much more sophisticated. But this is good for you to just look at a couple of things. As human beings, we have 22 autosomes, right? 1 through 22, and we have a sex chromosome right here. So the very last 23rd one is either an XX, you got an X from your mom and an X from your dad, and so you're a female, or it's an XY. And so in this karyotype, you can tell that this person expresses themselves on the planet as a male. So they got an X from their mother and a Y from their father. And you've heard of the company 23andMe? So that's where they get the number 23. Yes?
As a biology teacher, to put these karyotypes together, and I was doing some labs we did in
Yes. Never have. Let's do that. Let's do that at the next Heritage, one of the Heritage Hunter meetings. A little side table. Oh my gosh, I would totally love that. I think it's fascinating. That's so cool. Did you stain them, like with their staining involved, or do you remember anything? Oh, okay, gotcha. Very good. Okay, very good. Okay, so So you know that there's supposed to be 22 autosomes, right? They're called autosomes. And then that 23rd position is the sex chromosome. So there's 23 altogether. And you notice that there's supposed to be 2 at each spot, right? So just look at chromosome number 1, for example. This individual got 1 chromosome from his mother and 1 chromosome from his father. But at this point, from our position, we don't know which one, right? Like, we can't say, oh, that one's from the mom and then that one's from the dad. We don't know. They're pretty much, could be either one, right? So you can't really tell which one came from what parent, but you just know that there's 1 from mom, 1 from dad, and that's great. So the doctor that ordered this karyotype said, well, shoot, I mean, well, good. There's nothing, whatever's wrong with this patient doesn't seem to stem from a chromosomal imbalance or whatever. So let's go forward to the next one and just take a quick look along the chromosomes and see if you can pick up something goofy. Just quickly take a look. You want 1 from mom, 1 from dad, and just shout it out when you see it. There's 3 chromosomes at 21, and for some reason, this seems to get by our system of checks and balances biologically. So if somebody has 3 chromosomes at 21, it's called trisomy 21, and that will express itself as Down syndrome. But it's very common also to have trisomy 18 and trisomy 13, but those situations, the people rarely live on to adulthood, and oftentimes it's just kind of taken care of as a miscarry, in utero. Okay, so also somebody tell me one more thing. What can you tell me about this person? It's female. Good job. You guys are smart. Smart scientists. Okay, so this is the last slide on chromosomes, and then you guys are going to be experts in that. So we are rocking and rolling. This slide I really like a lot, and this is the first time that I start talking about genetic genealogy, which is what I do. But it focuses on autosomal inheritance, and I've been mentioning that word. The first 22 chromosomes are autosomes, right? So autosomal just means the information that you got from your parent, your 2 parents, and the information that they got from above, too. So the reason I like this slide right here is that it represents how information is passed down with color. So I like to think of this situation here as mine, because I have a brother and a sister. So I always say, this is Toby, that's Tom, and that's Terry. And then I just say that this is my mom right here, and this is my dad. So there's, my grandparents on my dad's side, grandparents on my mom's side, right? So if we just take a look at my grandpa on my dad's side right here, and just say that this is chromosome 1. Who knows what it is, right? But just say we're just looking at 1 chromosome. And then here's me down here, right? My grandpa, when his sperm met with the egg to make my father, there was a recombination event, and it did a little hopping. So my dad inherited a little mix-up, like a little mash-up of information from my grandpa. So there's a little bit of red, a little bit of blue that he inherited down from his father, right? And the same thing happened with his mother. She's represented with green and yellow. To form her egg, her body did a recombination event. He got a little bit of yellow and a little bit of green. So my dad runs around on the planet as a representation, and he doesn't really know this. He's actually not himself. He's like, actually those 4 people in a different combination, right? And so then he meets my mother, and they're going to start having kids or whatever. So I get 50% of my chromosome from my dad and 50% of this other chromosome from my mom. And you can see here how I'm actually not my father at all. What I actually am is all 4 of these, as all to just his mother and father, right? The 50% that I got from him, I got a little bit of this red, and then blue, and then I got that yellow and green, right? So my 50% from my dad is absolutely genetic material straight down from my And the other thing that I like about this slide here is that you can see now why my brother and sister and I, even though we're all full siblings, we are very, very different. So if this, for example, right, if this red tip that I received from Grandpa, if that were cancer, then me and my oldest sister were kind of the unlucky ones, right? But my brother's like, oh, you know, not me. But if it were something like red hair or whatever, that would like, you know, my brother wouldn't have that at all. He just doesn't have that gene at all, and we're completely different. But if we all did ancestry tests or 23andMe tests or anything like that, then for sure we would all come up genetically as siblings. You know, so the technology is good enough to see these differences, you know, but to know also on top of it that these three people are full siblings. So that is a really good premise stepping into genetic genealogy. Yeah.
I like this also for explaining, which I have trouble doing with people sometimes.
Okay.
And this doesn't show it, but you could do this to show it. Where you lose any inheritance from somebody
Yeah.
up here.
Yep.
You know, I know that my great-grandmother was half Cherokee, right?
Right.
And I get my DNA done, and there's no Native American in it at all.
Right.
Both of those things can be proved.
That's right.
Sometimes
You're right.
they're not, but
Yeah.
they
Yep.
can be, because with the successive recombination,
Yes. Yep.
something,
People can be just recombined right out of the picture.
right.
Yeah, exactly. Yeah, so what she's talking about, just to reiterate her point, is that sometimes if you go far enough, you'll lose a great-grandparent, or you'll even maybe lose a grandparent. It's not as common. But so say here's me, and if I go through a fancy recombination, and I drop that little yellow part on this side, then you know, I'm not going to pass anything on from this ancestor who was my great-grandparent. That yellow is gone. And so what she was saying was that if she, you know, if she takes a test, like a DNA test, and it's showing that she's not related to this person, it doesn't mean that she's not. It means that she's far enough away where it's been, you know, it's just recombined out. That's a good point. Okay, so now we're getting into a history lesson, a little bit of a history lesson here. These people, I just want to make sure that you all know about the players, like the people that were very instrumental in the groundbreaking work, you know, that was like fundamental for all DNA beyond it. And so that was Rosalind Franklin's crystallography work. She actually took a picture of the structure of DNA. It's called Photo 51. It's beautiful. And so she published that in the same Nature magazine that Watson and Crick published theirs in. And they were also, you know, they proposed the double helix, like kind of like that spinning part, right? So thanks to these guys, we got a really good start, you know, understanding, you know, the structure of DNA, which then allowed us to understand that it was kind of the heredity machine, you know, that passed down, that housed and passed down this really critical information from generation to generation. And then last but not least, he was after the fact, but Frederick Sanger is the godfather of sequencing, because what he decided to do in the 70s was look inside the DNA molecule. So Watson and Crick and Franklin, they were, you know, really busy just saying this is the structure. And then what Sanger did was like, what is in the structure? That is really cool. So Sanger he kind of gets the credit for being the first one to think about sequencing and really study those nucleotides, A, T, G, and C. I don't know. To look that up. Somebody can fact check really quick in the background. Okay, so fast forward, everybody, right? We just talked about 1977. This is how fast this lecture is moving. The year is 1984 all of a sudden, and we're going to talk about how law enforcement got involved with all of the stuff that we just talked about. Like, how did law enforcement step in? So in 1984, Alec Jeffries, who is Sir Alec Jeffries, had a laboratory and developed a scientific way of looking at a DNA profile called restriction fragment length polymorphism. So it was very soon after that called RFLP, and then everybody was like, that's even like worse or just as bad. So it was shortcutted to DNA fingerprinting. So that was the very first time that anyone in a laboratory could take somebody's DNA and look at it like on a piece of paper and compare it to somebody else's. You just do that side by side. So what he's holding up right there is a radiogram of actual pieces of DNA from people. Every row is a person. And he chopped up their DNA at the exact same spot. That's the trick. And then he ran the whole soup on a gel and realized that every single person had different chunks of DNA. Even if you cut it at the exact same spot, there was these different lengths. And so, right, yeah, right. No, it does, doesn't it? Yeah, it looks like a barcode. So anyway, he was busy like, wow, this is really cool. Published some papers on it, you know. And then a year later, this is how quickly they're moving, the police approached his laboratory and said, can you pick out a child from a family? And he said, yeah, I think we can. Yeah. And so what he did was there was a child coming back in from Ghana, and the authorities were saying, oh, sure, you know, you probably, you know, are you sure you're really, you know, a British citizen or whatever, right? So they took all of the family, including this child, and they ran the DNA fingerprint, and sure enough, he was a member of the family, and so they solved that case. So he invents it, they solve a case, and then the next year, 1986, the police are like, well, if you just solve that immigration case for us, can you solve 2 sexual assault homicides 3 years apart? And he was like, actually, we might be able to. So they tried it. And what they did was they rounded up the usual suspects. The police said, okay, if you're a male between the ages of, you know, whatever, 18 and, you know, 45 or 50, whatever, you are ordered to come down and give a DNA sample. And so Alex Jeffries' laboratory was quickly going through these samples to see if they could match the DNA fingerprint. And it's a very long and very interesting story, which you should look up after this. His name eventually was known as Colin Pitchfork. And they just identified it, right? Because they knew that they had a sexual assault homicide. And they compared the DNA sample from those homicides, and they knew that it was the same killer, same guy, identical DNA fingerprints. And so all they did was call these people down. Here's, you know, guy number 1, here's guy number 2, and they just gathered everybody and made them give their DNA, and they eventually found him because it matched. So that was 1986. And then fast forward to 1994, just because we don't have much time today. It's very interesting in between there. All of a sudden, DNA just really got accepted in law enforcement and became this thing where it was like, oh my God. And so law enforcement was like, oh no, we have to set up laboratories. And so they were really, really quickly setting up laboratories. And then in 1994, the federal government put through the DNA Identification Act of 1994, and INDIS was established. And that's and so now we call it CODIS, which is the Combined DNA Index System. But it was basically a database to collect all of these DNA fingerprints of all of these crime scenes. And then to start maybe collecting DNA from people that were, you know, in trouble and, you know, being, you know, like felons or whatever, right, for comparison. And so this was all started in 1994. But the one thing that they had to do was choose a platform. And this is where I skip ahead a little bit. So now in 1994, it's not DNA fingerprinting. By now, it's something called short tandem repeat analysis, or STR analysis. And what that does is that it identifies the section or loci on the chromosomes. And see how smart you guys are? You recognize these are the 22 autosomes, right? And then the sex chromosomes at the end. So this short tandem repeat analysis that they came up with really quickly in the 90s was just like, okay, locations and a DNA profile for that, create a database, and then start, comparing it to felons, getting in trouble for crimes. And then it worked like a charm. This is still what exists today, CODIS. Although today it's not 13, it was originally 13 STR core loci, but now it's up to 20. And so this is still what is happening at the Forensic Investigation Center in Albany and at every single crime laboratory everywhere. In fact, I have not heard of a law enforcement laboratory that is doing sequencing quite yet. So, but they're up and running, and I'm sure they're just like working like mad to try to get this going, right? But what I want you to know about this slide is that it's time for the new technology, right? We went from, DNA fingerprinting to STR analysis, but now we can, if you catch a weekend sale at Ancestry.com or over a holiday, you can, figure out, you can get your DNA sequenced for like, what, $59 or whatever, right? So we do sequencing now, and we don't necessarily do this anymore. So this is a bit of a, we're going to be bridging this gap in our society, but it is not bridged quite yet. This is becoming a little bit outdated. We need the new technology, but the law enforcement laboratories are not quite ready yet to do that. Okay, so the next two slides are just an example of how evidence flows through a laboratory, old style, and then I'm going to show you the exact same thing the new way that it can be done. Okay, so traditionally there will be DNA at the scene of a crime. Maybe right here it's just shown as a blood sample, right? So a swab is collected. It's wrapped up as evidence. Lots of recording. Chain of custody, perfect, and it's delivered to the laboratory, and then it's in the laboratory. It's processed. And the DNA is extracted from whatever source it came in from, like a tissue or blood or just whatever, right? So you extract the DNA, and right now, currently, we're still doing STR analysis in all of the crime laboratories. So the STR analysis continues on a genetic analyzer, and the profile is developed and put into CODIS, right? Just as I explained before. Since 1994, pretty much the same, right? So that's kind of like the circular way that DNA is used for law enforcement. What you do here in the CODIS database is that you have a DNA sample that was collected at the scene of a crime, and it's compared to a DNA database of bad guys. Every state's a little bit different, but basically the idea is up at the CODIS database that the feds run, there are a lot of DNA profiles from people who have been arrested or convicted of felonies, or in some cases convicted of certain misdemeanors, right? Every state's different. So we have a database of known people and then a database of unknown samples, and that's what's matching. And when I worked in California, I called them the year after I moved here to Saratoga Springs, and I said, how many DNA samples do you have now from convicted offenders? And they said over 3 million. And then I said, oh my gosh, that's amazing. And about how many hits, how many matches are you finding per month? And they said about 400, which is awesome. It's really good. So this system is great. So don't get me wrong. I mean, it's awesome. But yes.
So with CODIS, if you participate in like 23 or something, that gives your information elsewhere.
No. So this is the old-fashioned database. It's 100% law enforcement. The CODIS database is owned and maintained by the FBI. Yeah, good question. So back on your question, though, here is a modern picture using sequencing, right? Because we can use sequencing. People have Ancestry.com and 23andMe, right? Sequencing is around now. So can we use sequencing in place of STR analysis? And the answer is yes. And that's kind of like why I started my company. I was like, yes, oh my gosh, we have to start using this like yesterday. So notice, though, that it's the same 3 images, right? You still have the same crime happening. You still collect it and bring it to the laboratory. And in the laboratory, they still need to extract the DNA. But after that, it's crazy what you can do today. This is a genetic sequencer. Usually people have MySeq FXs. There's a few out there, but let me just toggle back and forth, right? It looks like a microwave. So did that one, or an oven, right? So you can see how easy it will be to either put these side by side or in one room and then another room, right? So that's what the laboratories are doing in almost like a panicked state right now. They're like, oh my gosh, sequencing is here. And the labs have been a bit blindsided. So you take that DNA evidence from the crime scene, and now you can sequence it, right? So you're looking for the sequence, the ATGC, for each person that left that DNA at the scene of the crime. So you run the sequence on the genetic analyzer, and of course, you get back a huge file, right? Because sequences are really big. Like the sequence of your DNA is like lots and lots and lots. It's not just 20 pieces of information. And then right here, these are the 2 databases now. So to your question, it's not CODIS anymore that can help law enforcement agencies. CODIS isn't ready yet. They've just been blindsided. They've just been like, what? We just got STRs up and running, and right? So these are privately held companies. GEDmatch Pro is owned and operated by Verigen, a company that bought over GEDmatch. And Family Tree DNA is privately held company. You can buy a kit. They're the only ones I think right now that are doing mitochondrial and Y DNA. So I've purchased kits from them before, right? But these 2 companies, thank goodness, each of them is like, we will do it. We will do it. We will allow law enforcement. Does Ancestry.com allow law enforcement? No. These are the only 2 on the planet that will allow law enforcement anywhere near their information. And that's important. Right. Great question. It's flip-flopped here. And they've done some interesting studies on this. When you enter your data into GEDmatch and upload it, you have to check a box that you want to support law enforcement. You want to help them. It's the opposite here at Family Tree DNA. When you put your DNA into that company, you would have to check a box like, get my DNA out. I don't want law enforcement I don't want to, be any part of that. Yeah. Okay. So now you guys are experts in all sorts of things. And if you want to think about why the law enforcement laboratories were kind of left in the dust, it's because of these companies. Nobody had any idea really how popular people learning about their heritage was going to be, and people love it. They just love it. There's over 20 million people at Ancestry. There's over 10 million people at 23andMe. There's millions of the other ones, although they're lesser than the top 2, but they're to blame. So they really did leave the law enforcement laboratories in the dust. So now we're left to scramble a little bit. Also, I like this graph because it shows what happens with the whole genome. And this graph is not talking about little like SNP profiles. I'll introduce that in a second. But this is like, how much it would cost to do the entire genome, right? So it's not that long ago that they first started, being able to map the genome in the first place, it's so quick, right? This is going so fast. And if you wanted to have your genome done, and it was like early 2000s, it would cost you $100 million. But people were so interested in this, so interested, and the grants were flowing, and there was academic laboratories, and there were research laboratories, and like all the laboratories in the whole world, right? Everybody's just, oh my gosh, this is amazing, right? So they just kept getting better. The instruments were smaller, the kits were better, and everything was just awesomer. And it goes all the way down to affordable. So that red line right there seems to be the price point. As soon as you got down to $1,000, everything changed and everything just really sped up. And so that was right around like 2015. And I mean, things just took off. People, just regular people. And mainly we have adoptees to thank. I have a slide devoted to adoptees that I'll show later. But they were the ones that were very curious. They wanted to know not only like their health panels, but they wanted to know what is their heritage. They kind of look a certain way. Is that because, their family came from here or there or what, right? So just people are innately curious about this topic. And I also liken it to this. If you're a music buff, then you will remember that right when it got to about the CD stage, you were like, okay, this is it. If there's one more quick change, I'm just going to, which was me. I loved my CDs. I had like a wall thing, like a shelf, and they were like alphabetized. And I don't have any CDs anymore today, right? That's how fast technology is. And you may hate it, and it may be, too fast for you or whatever, but there's no way of stopping it. There's just no way. And that's where we are with DNA and sequencing. Okay. Now I'm going to start to launch into genetic genealogy at this point. And before I do, I wanted to give a one-slide history lesson, because every once in a while I'll hear, it's not very often, but every once in a while I hear someone talk down to genetic genealogy a little bit and be like, it's a little bit hocus pocus, like, how can you really be sure that like all this or, whatever. But these were the early experts in population genetics, and with my training as a forensic scientist, I knew the Hardy-Weinberg equation. I had to know that, because this is where you get frequencies, allele frequencies in your population, which you have to know if you're going to run statistics on any DNA samples that you get. Right? So this is in the background and in the training, but for anyone that would want to start to challenge the legitimacy of genetic genealogy, all they would have to do is go back to Gregor Mendel or Charles Darwin and start the fight at that level. But since then, so many laboratories, so many publications, there's really just no questioning how much we do know about the flow of genes. There's just no question anymore. And also right here, when I talk to law enforcement agencies, I show this slide, but I say, don't you worry. Don't worry, but just know that you can trust the science. Like, this science is legit, and it's been legit for hundreds of years. And then I tell them that I'm the one that takes classes in this stuff. I'm the one that's getting good at recognizing endogamy versus pedigree collapse. How do you recognize it? And this is non-parental event, very common. And how do you, and this is surname DNA switch. So that's what I'm doing, and that's my challenge, and I'm learning how to do this, and I'm constantly taking classes and webinars, and there was a fantastic webinar yesterday through Legacy Tree, by the way, on Acadian ancestry coming down from Nova Scotia. So that's what I'm doing, right? So I tell law enforcement, you don't have to learn any of this, but you have to hire the right people, and you have to believe in it. You have to know that this is okay, and you have to take that jump, right? Just like the CD thing. It's a little scary, and you're like, no. So these are the modern experts. You guys may or may not recognize these main players in genetic genealogy, but this first individual here is Colleen Fitzpatrick. And then this is Margaret Press. Colleen is in charge of Identifinders International, runs that company, and she's a genetic genealogist. And Margaret Press heads the DNA Doe Project, and those people are incredible and have been doing really good work helping law enforcement agencies identify their Jane and John Does. Barbara Ray Venter is famous for the Golden State Killer. She was the genetic genealogist that helped that team of law enforcement individuals solve that case. And C.C. Moore also did, if you like to binge watch Netflix shows, I have a good one for you. It's called The Genetic Detective, and she did that. It's like 5 or 6 episodes of how she, as a genetic genealogist, worked hand in hand with law enforcement agencies and solved cases. Super awesome, especially episode 3. It's really good.
Why do you think women are dominating self-ED?
I have no idea, and it's happening in forensics as well. Like everywhere you go, the criminalists, the forensic scientists, they're predominantly female. I don't know. I don't know where the dudes are going. Maybe we're getting brave and we're like, we're going to be CEOs and stuff. I don't know. I'm like, I'm going to start my own company. I don't know. Like, well, but they have more patience. Okay, yeah. That's probably true, especially if they have kids. Okay. So this slide is, I make it terribly easy, but there really are only 4 steps to genetic genealogy. So here you go. Da-da-da. The first thing that you do as a genetic genealogist, if you want to find the parents of an adoptee or help a law enforcement agency solve a cold case, is you take a look at the DNA matches, right? So you send away the DNA to GEDmatch or FTDNA, or I'm sorry, to like Bodie or Parabon or some sequencing company, right? They take the DNA from it, they develop a really big sequencing profile, and then they send those matches to GEDmatch or FTDNA, and then you sit down at the computer. So you take a look at those DNA matches, and you're like, your fingers are crossed that you get a good one, right? And so the next step is that you pick your best DNA matches and start building family trees from them. And you ask who are their parents, and then who are their grandparents, right? And the third step is that you fit these family trees together. Up at the top, at the ancestral level, they will all start to share the same people. And you know that that's true now because you've seen it in the picture, and you've just seen these little chunks of DNA, and the DNA down low at the people that are matching, that's not a lie. If you have a DNA match, there is a reason, and it's a common ancestor. So you just have to find that common ancestor. And then the very last step is that you determine how your original sample fits into that giant family tree. So that's it. Easy peasy, right? Right. No, it's so hard. Oh my gosh. So this is an example of a case that I worked on, and this is just the step 3. So the step 1 was we took a look at the DNA matches, and step 2 was, we were like, let's build some trees, So I started with this kid right here, and you probably can't see, which is probably good because it's a real person, but this is all public information. So I started with him, and I asked the question, well, who are his parents, and I built that person in, and then who are these parents, blah blah blah, and then I gave up, and I was like, oh, this job is horrible, and then I was doing it for another DNA match over here. The pink is just an easy representation for me to female and male DNA match. So I was building a tree for her, blah blah blah. I got up to this top couple up there, and I noticed the last name Terrell, and I had this moment of like, oh my gosh, I recognize that name somewhere else, and I did, and it was over here. There was a Terrell that married into the Haynes line and produced that DNA match that went to Ancestry.com, and then that one went to Ancestry.com, right? So I started with those 2, build these trees, and finally found the way that all came together. So there are awesome tools to use today. GEDmatch, which I mentioned, has incredible, you can visually look like the karyotype thing. You can look at chromosomes. They have chromosome browsing tools. I think FTDNA has some too, MyHeritage, and for sure 23andMe. That's one reason to love 23andMe. And Ancestry has none, but they have really good family trees. So that's why we love that's why this last slide, that's why I build everything in Ancestry, but I've got to go outside Ancestry to do all the genetic genealogy DNA tricks, right? Okay. So always using Blaine Bettinger's shared Centimorgan project up there, which puts the person in the middle there, and then shows you what kind of centimorgan range. A centimorgan is the length of that gene, or that child, centimorgan. And so you're sharing genetic material with your DNA matches, and so it'll put you all, and it'll give you an idea. I think you guys are first cousins once removed if you're sharing this amount of DNA. If you're sharing these centimorgans. And then also, my leads team and I have been having a heyday with this tool down here. It's called WATO, What Are the Odds? And it's a way to what's that? It's a way to take DNA hits from any database and stick it into a family tree that you can rebuild pretty quickly with your Ancestry robust, tree building information. So you got to use the skills. You got to know about the skills, and lots of webinars all the time. And then this is an example of step 4, that last one, where you put the person. No. Oh my gosh. Okay, I gotta hurry. Can I have a time check? Somebody have the time? Okay, thank you. So this is an example of step 4. I had a client named Dave, we'll call him, and these were in red, those were his matches. So I built the trees up from his matches, and I found these people right up at the top. And then I was able to ask, well, now where would I put Dave? And we're looking for Dave's father, so where would we put Dave? And what I was able to do was rule out these 2 people right here. They were a bit too young. These ones were all kind of born a little bit too early, but this one was born at the right time to have children, to be Dave's father. So I gave that information back to Dave, and then he was able to take that and finish up finding out who his father was. Okay, so this is the slide where I thank adoptees for donating their very first instances. Back in the day when it was still pretty expensive, they were the ones that bought those kits because they wanted to find out who their parents were. So in the next 2 slides, I'm going to show you that finding solving a cold case and identifying human remains is exactly the same as finding an adoptee. So this is the scaffolding that you start with. An adoptee comes to you, and you look for the DNA matches, right, which are represented here as a first cousin, smiley face. So the question is, who are the parents, who are the grandparents, and then you just build it back down. What other children were at the right time in the right place to have given birth and give that child up for adoption. Look how similar it is. I just moved the slide forward. Adoptee, cold case. It doesn't matter, does it? DNA doesn't care. Look at this. Human remains, right? So if you have these 4 skills, it doesn't matter what kind of case you're researching, whether you're working for your family or friends or a law enforcement agency. Same thing. Okay, so here's the bad guy, and this is the case that I'll breeze through real quick. And this was in 2018, August 24th, 2018. I was in Aunt Julie's kitchen when he was arrested, and I will never forget that. This is a picture of me working in California in the DNA database vault. Here's where the 3 million samples are housed. In California, we were all embarrassed. It was horrifying that we could not solve this crime, that we couldn't catch this guy. It had been, at that time, going into the 4th decade, and we couldn't find him. We did crazy things like the Proposition 69 was passed, we collected all arrestee DNA. Anybody who got arrested for a crime, for a felony, not for a misdemeanor. But I mean, to some people that's even, a kind of, severe. But we were willing to do anything to try to catch this guy. And then we started the D3 program, which was the Deceased DNA Databank program, because we thought he may have died in prison before giving us a sample. So this is the team that was formed after 4 years when we couldn't solve that crime. It was led by Barbara Ray Venter, who was a genetic genealogist, and somebody had the idea, actually it was Paul Holes right here. He had the idea in 2017 to try this on the rape sample. So what did they do? They did what we do. They started researching archives, census records, obituaries, blah, 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 everything. And then they also got very lucky with an X match, which is helpful in DNA. But the team of 5 people just worked and worked and worked. And they came down to this. This is actually a snapshot. I was in a webinar, and I was like, snap. I snapped this, but it's okay. I'm sure it's okay that I have this. But this is this is Steve Kramer's presentation. I'll give him credit. It was Steve Kramer's presentation. But this was actually them working in Ancestry.com to find the Golden State Killer, right? So they started with a sample sexual assault from the California area. They entered that, had it sequenced, put it into GEDmatch. I think it was just GEDmatch at the time. But then the challenge became, okay, build the family trees, and they only had a 4th cousin match at the beginning. So they had, that was really challenging for them. When they finally built all of the possible trees, they realized that there were 6 people. It went really fast at the end. There were 6 people that could be this guy. So what they did was they looked at Prometheus, a database in GEDmatch, I don't think it's there anymore, and confirmed that this individual had blue eyes. Then they went back to the case records, the victims that did survive, every single time, blue eyes, blue eyes, blue eyes. And only 1 of the 6 possible suspects had blue eyes. So Barbara Ray Venter turned over 1 name. She just said, here's your guy. I love her, she's so bold. She's like, here's your guy. And they're like, what? Are you kidding? She's like, yeah, no, I'm not kidding. There he is. Go get him. And it was him. So in these last 2 slides that I have here, this is just the numbers for traditional investigation. It was $10 million, 43 cold years. It involved 15 law enforcement agencies, 650, I forget what that number was. Probably, oh, thanks, Dawn. Dawn's on my leads team. Thanks, Dawn. And 200,000, Dawn, do you remember that one? I didn't bring my notes. No, so it was 300 people had DNA, like they were surveyed, 300 people surveyed and DNA collected. Anyway, all this stuff, right, it boils down to we never found anybody all that time. If I had still stayed at the laboratory and we still were doing STR analysis just like they are, we still would not have found him. They still would not have found him. It was only through genetic genealogy. So these numbers I do remember. This was $217 to run a SNP, the sequencing for the kit, was those 5 people, and it took them 63 days to come up with 1 lead. And so, Dawn, here's Dawn in the back, present today for the talk. Nora is not here, unfortunately. She's our team trailblazer, and Kevin Sullivan is a retired investigator. But these people they just reached out to me in one or more ways. Email. Nora took my class, just and said, this is incredible and crazy, and I want to be involved. How can I help? I think Dawn, sorry to put you on the spot, but I think Dawn said, I know I'm a little too old to be an intern, but I was like, you know what, maybe not. But this is great, though, because They, for those cases that are going to go on for a long time, when we don't have really good matches, or we have endogamy, or we have non-parental events, these people, we all just put our heads together, and so far have solved, I'm going to say 2, because I think you have solved that very last one. And a third one is coming. So that's the leads team. So I'm not alone. It's not just me, these people. So now I'm just giving you kind of closure slides about some things like that I'm doing, involved with, or whatever, the community. I have internships, and so I've been carrying 2 internships since I opened up my company, and I've covered colleges and high schools, and in the summertime there's more time, and so those internships are intense and good. They're awesome. One of my interns went around and filled Find a Grave requests. So this was just one of and she's a great photographer, and so her Find a Grave entries were like, wow. Yeah. So, and then we had another intern that set this website up. It's called DNAlookout.com. And if you go there, like on this first tab, for example, unidentified human remains, if you click on that, it pulls up a state of New York, and you can click on a county, and it will show you all of the unidentified human remains cases for that county. Because a lot of times the law enforcement are kind of like, oh, I didn't know you, if I find one, and I'm like, did you know that you have, and they're like, let me get back to you on that. So anyway, there's a lot of really good work going on. And I think here, so the summer internship is coming up, and I'm entertaining applications from Siena and also the New Jersey Institute of Technology, where I just received an invitation to be an adjunct teacher next spring 2023. So I'm really excited about that. But this is my really unapologetic cry for help today, all right? So this is where I need help, and I need if you have advice or if you have ideas, please get my business card or just catch me or Dawn, and we need help. This is my slide, and I'm going to show you where Saratoga needs help right now with genetic genealogy. So this is my reality pie chart, right? And I'm doing this cold case one right now, and it's working pretty well. I've got the ball rolling, I've got clients, I'm always busy, blah, 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 it's rolling. But there are 2 pieces of my pie that are infuriating and really sad, and genetic genealogy hasn't even touched it yet. And these types of crimes that could really use genetic genealogy are hot cases, which are cases that, why wait until they're cold, right? So you know how we talked about CODIS and delivering a sample to the laboratory? Well, there are laws now. Part of the backlog reduction law for the state of New York was that agencies have 10 days to deliver a sexual assault kit, right, and to the laboratory, and the laboratory has 90 days or something like that, right? So everybody has a finite amount of time to really move these cases through. But one of the problems is that after 90 days, if there's no hit in CODIS, there's this drop-off into nowhere land. And so I've come up with this little hashtag meme kind of thing that I've been promoting a little bit. Maybe I could get a summer intern to pick it up. But Project 91 is kind of like start genetic genealogy the moment that you hear back from CODIS and there was no hit. If you hear back from CODIS and there was a match, that means that there's a lead for the law enforcement agency. But if the CODIS testing comes back and you're empty-handed as an investigator, you can do genetic genealogy. And it takes us between 45 minutes and 2 months to solve a case. If you have DNA, basically we're solving your case. That's how it is today. That's how just succinct this science is. There are some cases where you can't, or they're difficult, or you have to wait maybe, but we haven't run into them yet, right? So I'm just going to say that these are solvable. And then this last little chunk here, we need a lot of help with. We need a lot of help, and this involves policy. In the state of New York, we can't send forensic. You can't send a bone. If you bone or, like a femur or a mandible or a tooth, right? You can't send that to a laboratory that is not Department of Health permitted to receive medical specimens. And so that's a bit of a hang-up that I would like to see if there's a different way around that. It's just the way that New York State kind of packages forensic specimens. It's right under the umbrella, goes right along with all of the hospital samples and stuff like that. But that puts a lot of pressure on any laboratories that exist outside of the state of New York that actually do sequencing. It's too difficult to apply for the permit. No one has done it yet. None of the Doe cases that exist in the state of New York can be solved right now with whole genome sequencing, which is what they need. Whole genome sequencing is like big, it's like the whole thing. So that's what you need when you have bone and teeth. So anyway, that's a problem because if I had the sequence, I could solve the case. I could deliver back to the agency the name of that person, and that would just close it out, right? So that's a good thing. So I'm working on this. So tell me if you have any ideas there. And then the last thing that I need that you can help me with, give suggestions or guidelines, is funding. I have a case right now where there was a homicide victim found in Lysander, New York, and the grant company that I usually use, Season of Justice, and they're great, they're really good. They will not do adult Doe cases. They will not fund. I can get into why, but they won't. And so this poor investigator who wants this crime solved, I didn't put the date on there, it's 70s or 80s. I can solve this case with my team in 2 months, as soon as we get $6,400. And so it's funny because, and if you're wondering why it's different than this, it's because they collected a blood card, and they collected a lot of, so we don't have to do the whole genome sequencing right up bone. Okay, anyway. So so I'm looking for like creative ways to fund these cases. And the investigator's like, what do I do? And I'm like, I don't know, let's try something. So we're coming up with fundraising ideas and stuff like that. And we want to get really creative. And a lot of times if you go to a foundation and say, hey, do you want to solve a crime? It's going to cost, $6,400. They'll probably say yes, right? I mean, that's a really cool thing to be able to say, I solved this crime. Oh my gosh, thank you so much for waiting with me through the whole thing. And I will entertain questions now. You have them. Thank you for coming, though. Sorry I went over.
Have you tried GoFundMe?
No, but I could, right? It's funny, there is something called Doe Fund Me that someone came up with. It's Colleen Fitzpatrick. So, but yeah, why not, right? Right, why not? Yeah.
You spoke on Ancestry.com having really good family trees, and I wasn't sure what you were referring to. I presume
Yeah.
not public member trees.
I am referring to that.
Are you?
It's the best.
But
Yeah.
so many of them suffer from a lack of understanding of geography.
Oh, yes.
They say so-and-so is the same person
Yeah.
who was born in California and died in Scotland.
Yeah, I know, it is true. So just to repeat, his question, he's asking about the trees of Ancestry and how I mentioned that that's our go-to tree building outfit. And right, the hints are very good, right?
I don't use this. I use the trees to find
Yeah.
people, and then I evaluate whether they knew what they were doing.
Right. Well, in my business, what we call, and you can look this up, and there are little webinars on it. It's called quick and dirty tree building, and we're getting good at that. Like, we don't actually care about the real dates and stuff, But what we want to do is follow the DNA path, and we want that big family tree, and maybe the spelling is off, and like all this kind of stuff, right? But it's good enough for us for quick and dirty Q&D tree building.
Well, and if you have been doing genealogy
Yeah, okay. Looking good, looking good.
But you look at the tree and you say, oh, the child was born before the mother was born.
Oh.
I don't think so. I don't think I'll use that tree as a source.
Right. Yeah, yeah, there are those, yeah, isolated,
And then
right.
somebody else, there's 7 of them with the exact same error.
Yes, yes, yeah. So her question is, do you still have to deal with the, mining through the mistakes? Yes, you do. Yes, really. Yeah, yeah. Go ahead.
Yeah, so I'm really cooked on this DNA. I really enjoy working with it. I do it a little differently.
Have you spoken on it before?
Well, for the heritage.
I knew it.
Yeah.
I was at that. I wrote your name down. Anyway.
But I've been able to find for adoptees. I found 2 or 3 biological parents for them.
Great.
A number of brick walls. But I use Ancestry, which already interprets a lot of DNA for you.
Yeah.
And matches. And I use triangulation.
Yes. Which is why I liked your lecture.
Because I don't understand all the sequences.
Oh, really? Oh, okay, right. No, but it's the same thing. That's it.
And then
Yes.
documents, and then historical stuff that
Yeah, yeah.
fit it all in.
Yeah, so just to reiterate, his comment was he wasn't exactly seeing genetic genealogy flow through like he did it, but he's using much more of the just ancestry with triangulation. And I usually have a slide on triangulation. I have a longer training that I do for law enforcement agencies. And I bring that up as another, don't forget, lesson that the transitive property of equality has existed in mathematics and biology for those, hundreds of years. And if A equals B and B equals C, then A equals C. And you don't really have to go through the proof. You can.
I actually found out that because of the DNA match, I found out that one of my first cousins had an illegitimate child.
Yes. Yes. He's commenting that there are surprises when you start opening up the Pandora's box. Yeah.
When you're working with families, trying to find from them who's your siblings, who's your parents, so they sometimes just say, I don't know, or I'm not going to work with you.
Yes. Just this morning, I was counseling someone who's, she's actually working with me. She's interested, with our leads team. And, it was so awful because she said, Toby, they didn't want to talk to me. They thought I was a scammer. And I was like, I'm so sorry. I know it's going to happen. So, but it's interesting. She told me about her conversation with this woman that she just cold called, right? And they're talking about the same thing. Oh, I had breast cancer too. And oh, yeah, that runs in my family too. And so I'm like, come on, people. You do need to talk. You should really talk. They're like first cousins or maybe once removed. Yeah.
Related to the not talking about things, I'm Swiss on my father's side and American on my mother's side. And I have 16 or 17 first cousins in Switzerland.
Oh, wow.
And I did my DNA ancestry. And I got in touch with one of my cousins I know, and has anybody in the family over there done anything because, it opens a whole
It does.
realm of things on my father's side. And she said, oh no, we don't do that. We only do that to find out that our father is not our father. We are all Swiss, she said, but we share a Dutch great-grandmother and a German great-grandfather. She and I.
So they were Swiss.
They're all Swiss.
Yeah.
I was hoping for, the Dutch great-grandmother to have some exotic entry that didn't show up in mine.
Right, yeah.
Because so many
Wouldn't that be
ethnic
cool?
groups went through the Netherlands,
Yes, yeah,
I
yeah.
could
Yeah.
Or,
Yes.
no.
I know. I'm 1% Nigerian, and I love it.
Oh, yeah. No. I'm
Totally awesome.
93% Western Europe and 7% Scandinavian. That's just the Vikings in England.
Yeah. Right. Right. I know. Is there a question? Yeah. Go ahead.
Unlike so many of the other stories didn't know anything. I assume from popular knowledge that when you do DNA, you take the DNA of the suspect, and it matches the DNA found on the fetus file, it doesn't match. Is that not what's going on in this file? I mean, to get catch the person, what they're doing before them. Because you know,
Right.
what they're doing, why.
Yeah, actually, so I didn't cover this today. So his question is about kind of driving home that the point at the end with the DNA in court and the part of genetic genealogy in that. So just briefly to cover that, the role of the genetic genealogist is to deliver the lead. Like in that case of Joseph James D'Angelo, they turned over one name. But what they did was they said, you should look into this guy. But in the genetic genealogist's head, we're like, that guy did it, right? But what they still have to do, the law enforcement agency takes the information. Oh, you say it's Joseph James D'Angelo, do you? And then they trail the guy. They survey the individual. They collect a surreptitious DNA sample or a trash sample, a trash test, right? And they take that sample and they run STR, old-fashioned analysis, and they compare it back to the sample from the scene of the crime. And that's what moves forward in court. Genetic genealogy has really yet to enter court.
The other question is, you're not involved with the trial at all.
Right.
Because that's where it's a very problematic issue. If you make a whole expert look like a fool,
Right.
it's like it's really nothing to do with
Yes. Right. Yeah.
what the jury knows and what
Yeah. So he's asking about expert testimony. And as of yet, genetic genealogists have not been pulled in. There's one case that you can look at. I actually have the transcript, and it was the NorCal rapist, where the defense really tried. What is this hocus pocus? Genetic. We need to see all this information, and the judge was like, "No, you don't. It's pretty much like a tip from a hotline. We just say, if I were you, I would, based on what I see, looking at everybody's DNA, you should go look at this guy." And then they do that old-fashioned collection process, comparing brand new, totally, brand new, unsolicited DNA, modern day DNA, and comparing it back to that, and that's really what goes to court. Yeah.
But one of the things I'd like to suggest is that in families, just like that, whatever, if you think of it, simple as your life. When my mother died, I found out what her breakthrough. I found out how to understand still. Well, my real father was Donald Rose.
Mm-hmm.
I could show you the picture. I looked at it like
Wow. Yeah.
everybody knew about me.
Right. Oh, families. Right. Right. So she's issuing a cautionary tale of moving forward. Everybody should take a piece of, a swab, get some cheek cells, piece of hair, piece of clothing, put it in an envelope and stick it in the freezer and forget about it, their name on it. Excellent suggestion. Yeah. Yeah, no, So I think if this is right, I'll reiterate your question. When you're watching the flow of the sequencing, is there any predictable pattern with like traits being distributed down? And I think the answer for me is no, I haven't seen anything like that. I haven't seen anything that has, you know how patterns are, they'll pop out at you. They're repetitive. You do the same thing and you get this one thing, right? It's so random. Every single event. And when you dive into meiosis and how, you know, the reproductive organs make sure that the daughter cells are completely unique, they are all different. Every single egg is different than every other egg. And every single sperm is different than every other sperm. Except when you have twins.
You can still have 4 generations of women with the same eye color.
Yes, and right, you can. You can have, everything can be conserved, and they can be red hair, or totally different. I have a friend that has an African-American husband. She's Caucasian, and they have one black child and one white child. Yeah, and they both look just like them in the facial features. It's great. They're beautiful kids, too. Yes?
How many genes are in each individual's genome? And second question, if you ever look at Carl Lewis, that had little case that went to specific conditions.
The second part was, have I ever looked at Louis Gates? Oh, I watched an episode last night. It's really good, yeah. And I think he employs the C.C. Moore as a genetic genealogist, yeah. Yeah, I love that show. What was your first question?
Well, just how many genes are in every cell, every organ cell?
Oh,
What is the number of genes?
right. Just a few weeks ago, we finally finished the entire genome, right? Did you see anything about that? Oh my gosh, as if we missed it? I don't know. There's so much there. It's just a vast universe. I am not sure how many genes are in the genome. And I don't know how anyone would answer that. That's a great question. But maybe they're about to, finish that up now when they can say, okay, whew, we got through it all. It's all sequenced. So now maybe they can go through and map. Of course, once you think you know something, you start to realize you don't know, right? Because we chose the STR analysis, we chose those STR regions initially because we thought they were like gobbledygook, and now it turns out that they actually might have a role. So that's okay. That's all right. Yeah.
Do you have any advice for very young person who's going off to college and wants to go into the field?
Yeah, so the question is advice for youngsters. I think this is yes. Oh my gosh, this is a good one to go in too. Lots of future possibilities everywhere. I was just reading an article about the youngest cold case investigator. She's the head of her cold case unit, and she's 20 years old. There's an article out about her. If you want to contact me, I'll find it for you. And I was like, man, her retirement is going to be awesome. Can you imagine? If you started working when you were 20, your benefits would be like, whoa, so great. But the sciences are a great place to go. And the companies are really cool. There are companies that are really getting behind my business. One of them is Offram. That company is in Texas. They are all about whole genome sequencing, SNP profiling, and any other kind of sequencing you might need. Hair is really cool. They're doing all of that for law enforcement, nobody else. So I'm like, yes, it's great. So I mean, the sky is really the limit, and yeah, it's a good field. It'll be change. If you're ready for change, you got to be ready for change. You can't hold on to those CDs and be like, oh, my CDs. Yes, yeah. What are you going into? What grade are you in?
I'm a senior going to college next year.
Nice. Okay. Yeah. Definitely get my card then. And reach out. Yeah. Yep. My summer internships are open right now. No one haven't even started killing them at all. I'll start that in May, toward the end of May. I'm taking an endogamy class in May. Okay. Was there a question over here? No. Okay, are we wrapping things up, maybe? Okay, thank you so much, everyone. Thank you very much.